Let me start today with an unfortunate fact. Agriculture is bad for the climate. Cows and rice emit methane. Fertilizer use results in nitrous oxide emissions, and taken together with things like deforestation, there are publications that have reported close to 1/3 of all anthropogenic greenhouse gas emissions stem from agriculture. At the same time, climate change is also really bad for agriculture. More floods, more droughts, higher peak temperatures, and more pandemics, and not just pandemics for people, but pandemics for plants that could ultimately result in widespread famine. Both of these problems, ag emissions and crop loss due to climate disasters, are just going to get worse as climate change intensifies and the world's expected to add close to 2 billion people in the next 25 years. How are we going to feed close to 10 billion people without destroying the planet? What if I told you we could create a farm that doesn't have any net greenhouse gas emissions? A truly net zero farm. A world where we could grow all the food that we need, even in the face of a changing climate, and be part of the global climate solution. I'm here today to talk to you about CRISPR and how it could do exactly that. I'm the executive director of the Innovative Genomics Institute, the IGI. We're a nonprofit biotech hub on the campus of UC Berkeley. And we were founded by Jennifer Doudna, who won the Nobel Prize for her work in developing CRISPR, a tool that scientists can use to directly edit DNA and change the function of living cells. CRISPR is two parts. It has a precise targeting molecule called a guide RNA that allows you to be able to zero in on a specific part of the DNA. And the second part, that's a molecular scissor called Cas9. And when you take these two parts together, you're able to zero in on a specific segment of DNA and cut to inactivate a gene, or even paste in DNA segments that can change and correct a disease-causing mutation, or change the trait of cells like plant cells and microbial cells. Now, last year was a big year for CRISPR. Got the first FDA approval for a new CRISPR drug that essentially cures sickle cell disease. It's really remarkable. But what excites me about CRISPR is that it doesn't just have to be relegated to creating new medicines. DNA is in all living organisms. It's the code of life. So why not unlock the power of CRISPR to be able to solve other major problems facing society? And one of those problems is the need to be able to provide food security into the future. IGI scientists have already been working hard on this problem. We can edit over 30 different crops: rice, wheat, sorghum, tomatoes. We can create pathogen-resistant crops by knocking out disease susceptibility genes. We can create drought-tolerant crops by using CRISPR to reduce the number of water-losing pores on the leaves of crops. We're even working to save the banana and chocolate from emerging pathogens. And I promise you, in the coming decades, farmers are going to be using these crops to be able to increase yields, to reduce crop loss, and basically grow more food using less land and less resources. But to make the biggest impact, we also need to use CRISPR to be able to directly try to turn off some of those agricultural emissions. And those emissions, surprisingly, the most of them don't come from tractors and burning fossil fuels. Often they come from tiny microorganisms that live in communities called microbiomes. And those microbiomes are found in places like the gut of a cow and in the soils that we grow crops in. And they result in the emission of potent greenhouse gas into the atmosphere. Now, the worst of these emissions come from cows and livestock. Depending on what publication you read, anywhere between 10 and 20% of all anthropogenic greenhouse gas emissions stem from livestock. Now, our collaborators at UC Davis have actually developed a feed additive based on red algae seaweed that actually shows that you can shift and change the microbiome to reduce the amount of methane that's emitted. But it's really difficult to scale and sort of grow around the world, and you have to give it to the cows every single day in order for it to be effective. We're working on a CRISPR treatment that you could potentially dose just once, right as the calf is born, when almost every calf in the world is handled. So we really think we can actually shift the microorganisms to a low methane or even zero methane state and reduce those emissions throughout the entire life of the cow. Now, that brings me to our plan to be able to try to absorb gigatons of atmospheric carbon and put it back into agricultural soils. And we think those soils are a perfect target because they've lost close to 500 gigatons of CO2 equivalents. What if you could use CRISPR to help put that back? Our plan is in 3 parts. We want to use CRISPR to enhance photosynthesis. That'll increase the amount of atmospheric carbon that you take and put it into the biomass of crops. But you can't just stop there. It's more carbon for food and more carbon to store, but a lot of it would just be degraded and put back into the environment. So we're going to try to use CRISPR to put it into more stable forms, like deeper and more dense roots. And the deeper you can put carbon, the more likely it's to stay in soil. And then work with the soil microbiome to try to lock that carbon into nice, organic, fertile soils. Now, we all know the scale of the problem that we have. It's huge. And people are turning to direct air capture, and it's important. We have to do all of these things, but it's energy intensive and difficult to scale. Meanwhile, I tell people that biology already scales, and agriculture means you can put seeds in the ground each and every year and affect change really quickly. So you can see why I'm excited to be at the IGI and realize my vision where global climate emission goals can be met by changing the way we grow food. And it comes with so many different co-benefits: reduced emissions, less atmospheric carbon, more food security, and enabling farmers to grow on marginal and underutilized lands and convert them into more fertile soils, reducing the need for deforestation. So let me leave you with one word, and that's hope. It's easy to lose hope when it comes to climate change, but I'm here to tell you we can make a difference. Scientific innovation can make a difference, and together we can build a more sustainable future. Thank you.